Good day. I would like to start with a formal apology that I cannot be with you today in person. I'm Gerda, and the aim is to give you a brief overview of pest management in South African macadamia orchards. Starting with just one slide, and what I briefly want to illustrate is the incredible growth that we've seen in production in South Africa, with growth that we know will continue as more orchards come into full production. But with the positive, we also need to look and consider some threats. Threats to production, that is both in terms of yield as well as nut quality. And in South Africa, we have a growing list of pests and diseases of concern. But the focus for today will be to briefly touch on our insects. Before I get there, just a general overview of our research program at FABI and the University of Pretoria. Here we say we work to reduce the impact of pests and pathogens. We want to think solution orientated, so what can really help the farmer, and all of that sort of comes down to improve management practices. So we often focus on pest or the disease life cycle, but also how it interplays with your environment that we overlay with tree phenology. This is to ask the question, are there specific stages that we can identify that we can then target towards control? To start off with stink bugs. Of course, Bathyculia distincta, the two-spotted stink bug, is our most dominant species, but we know that there's at least 20 species that can be present in our orchards. Growers today mostly control these species by means of insecticide sprays. However, some of the research that we did recently showed that pyrethroids are slightly more tolerant than the organophosphates. But perhaps more important is what I've listed here at the bottom, is that we could show that these sub-lethal concentrations can actually increase dispersal and it can accelerate the development of their offspring. So our results with the negative impact of using insecticides, as well as a number of products that we know are going to disappear off the market quite soon, really should motivate growers towards alternative control practices. If we summarize integrated pest management, it is starting at what is preventative measures. I know this is something that Jeremy definitely talked to you a lot about, and thereafter move into biological control as well as chemical control. But to know when to apply a control measure, we need to understand economic damage. And for that, scouting is crucial. But something that can also be used in addition to scouting is making use of prediction models. So here, for example, Mulalu, who was a PhD student at the University of Venda, developed a degree day model. And at this point, SAMAC has now taken this degree day model and they're busy putting it into the SAMAC integrator. And the aim is before the end of the year, you as a grower would then be able to use and log into the SAMAC integrator, add your orchard climate data, and this app will help you to understand the number of generations of stink bugs, and so also can be used as a warning. So with scouting and prediction models that we hope will be available soon, you can have a firm understanding of the number that are in your orchards. But some questions remain. And so our question is, where do they come from? What are these alternative hosts that they can breed and feed on when there's no nuts in the orchards? Now, so for this, we took a study where we collected some stink bugs, we dissected their guts, that is in the picture in the middle, and we then extracted DNA where we specifically focused on amplifying plant DNA. So we wanted to know what were the stink bugs feeding on prior to arriving in the orchard. So what do we think and why is their value? Well, we hope that we can then identify specific host plants that growers can use as sort of early pest monitoring. Or if we have some of these alternative plant hosts, we can maybe add the alarm pheromone that a student in my group recently characterized, and we can perhaps develop the type of push-pull system. The following is then an abundance curve. In other words, the amount of other plant species we detected in their guts. Yes, this is a complicated figure, so in the next few slides, I will simplify. But first of all, what I want to point out is that in these different growing areas, we have an early and late sampling. Our early samplings is just when we started seeing nutsack, when the first stink bugs arrived, and our late sampling is just before harvest. If you compare, for example, 2022 KwaZulu-Natal early versus late, we see that we pick up more plant species in the beginning. These results suggest that yes, after the winter, they move in. But because we also see some other plants later in the season, although less, we know that they can breed and they will establish in the orchard. But we can also see continuous movement from stink bugs from the surrounding areas into the orchards. And that is something that really complicates management. Even if you do regular scouting, you never know when new ones will arrive. As promised, the previous figures simplify. In total, we detected 35 different plants in the guts of stink bugs. Now that is actually an incredibly high amount because our survey only allows us to detect whatever they feed on within a 48-hour window. But what did we find? We had 14 species that are all commercially grown. Of course, macadamia were there in a very high abundance. This is because we sampled them from macadamia orchards. But some interesting results was, for example, that in one of the seasons we picked a high number of green tea from bugs collected in an orchard in Mpumalanga. And we know that there's a commercial farm about 35 kilometers away. It also just tells you how quickly they can travel in 48 hours. However, in the next season, we didn't find any green tea, but we picked up quite a lot of cannabis. And we also know there are some commercial farms in the area. But one crop that stood out was pine. We picked up pine early and late in the season, and we picked up pine in all three our growing areas. Now, this result is interesting because recently it was shown that the brown marmorated stinkbug actually uses pine as an overwintering host. So this is something that we really need to look at in more detail. We then also detected 9 native species, some of these species that might be their original plant host. And then finally, 10 sort of known non-native species that we also know naturally grows in South Africa. So from this data, we have some homework still to do. Our plan is then to provide these lists to our growers so that they can look out for these plants in their surrounding areas. And we hope that they can help and we can set up some early monitoring. So understanding stinkbug movement, the surrounding environments is a key aspect. But we of course also needed to really say which stages in their life cycle can we target. And in the following few slides, I'll go into more detail. So firstly, EPFs or entomopathogenic fungi. So here the target will be adults or some of the nymphal stages. The figure that I'm showing you is then just some results of treatments where we incorporated and used commercially available products. The red line is A, that is our control, so stink bugs that were not treated. And you can see that after 10 days, the population is still doing very well. And so what we can detect from this slide is that all the commercial products that we actually tested can work. So this is definitely an option for growers. And our results really complement some of the work that has also been done by other academics sponsored by SAMA. First of all, that was an MSc study by Lungile Linda, where she focused on finding indigenous strains. And she actually got 100% mortality. It worked better than an insecticide spray. Then at the bottom, more recently, an MEC study that was shown at the University of Venda used a combination of products. So here he added Bavaria and Metrorhizium that are then EPFs because they're fungi in combination with the bacteria that is Bacillus. And they also added some chitinase, which is an enzyme. And what they found in the field is that the EPF treatments were actually similar to a knockdown spray. So once again, results that really show that these products can work very effectively. But then moving on to a different life stage, and here specifically, what about stink bug eggs? So first we need to talk about egg parasitoids. So Tosorcus bisalis is a product that's commercially available in South Africa. And so again, work from Sean, that's the student at University of Brenda, really showed that it can effectively parasitize Bathyculia distincta eggs. However, during our surveys, we actually collected a native telenoma species, and that species now needs to be tested. So our plans moving forward is to really look at the telenoma species, comparing it against Drosarchus, and maybe adding another parasitoid that growers can use. But what else when we think about eggs? Well, here we want to use a specific and interesting life aspect of stink bugs. So we know that the guts and the bacteria in the guts are really, really important for stink bugs. So too is the gut biome for humans. And what we know is that when the female lays her eggs or oviposits, she actually smears that layer of bacteria over the egg. And when those nymphs hatch, they actually consume their gut bacteria. So we conducted some trials in the lab to see if we can take freshly laid stink bug eggs and treat them with the aim of removing these bacteria from their surfaces. Now the following graph is then just tracking mortality of the stink bugs using different treatments. First of all, you see the green line, that is our no treatment, where we just simply allowed these eggs to hatch and we track development. We then have a 70% ethanol, which is the pink or red line, that we use as our positive control. We know that ethanol will definitely remove the bacteria, but of course that's not something that you can use in an orchard. So we had then three different treatments. First of all, just dipping them in distilled water, and then we used two commercially available micronutrient products available, Verno Trio, Verno Copper, and Zinc. And our reasoning behind using these products is first of all that macadamia growers already use these products from time to time. And we know that especially the copper, the zinc, and the manganese have antibacterial properties. So if you now look at the pink and blue, which is then our treatments, and you follow the graph, you'll see that there is a huge difference between our no treatment and our treatments. Interestingly enough, we also saw disruption of life cycle and a reduction in a huge dip in mortality when we look at the distilled water. But here we think it's because we used freshly laid eggs. And that matrix where that bacteria survives takes up to 48 hours to develop. So if we then had to repeat the trial and we used 2 or 3-day-old eggs, we expect to find that the water treatment is not so close to our actual treated Verno Trio and Verno Copper Zinc. The following graph is again the same treatment that I showed in the previous slide. It's just displaying the results differently. Here we tracked and we wanted to see how many of these nymphs that hatched actually reached adulthood. And you can see in our no treatment, a lot of them reached adulthood. If you look at our 70% ethanol, you see that none of our nymphs actually became adults. And now perhaps it's easier to see that there is in actual fact a difference between the Verno Trio copper zinc manganese and your Verno copper and zinc compared to the distilled water. But as I just said, yes, because we use the freshly laid eggs, the water actually also removed bacteria. But this is promising results, and it really shows that if you have a high number of egg packages in your orchards, trying to treat and remove that bacteria before the nymphs hatch is something that you can use as a control measure. I also briefly want to touch on another very important insect, and that is Macadamia felted cosset. Yeah, I've included some pictures of it, and you can see it's a very, very small scale insect. Of course, it's native to Australia, but since it's been introduced into South Africa, it's causing major damage. And here I'm just displaying, if you can see, the scales on nuts, on the branches, and if it's a heavily infested tree, you can actually see the tree dies. So what is our current research? We're currently again working towards a degree day model. So we are asking questions on exactly how long it takes from eggs to adults at specific temperatures. So this is the one that was already available. It was published by researchers done in Hawaii. And we are very close to being completing these other temperatures. And the value of that is that we would once again have a degree day model or a prediction model available that we will then share with SAMAC. And that can then be something that the grower can help to predict, to understand how many life cycles can he expect in a season. And it can also help him to understand when the population is peaking. However, since this insect, MFC, has only been introduced in South Africa a few years ago, we also did something that is called ecological niche modeling. So here we worked with researchers in Hawaii as well in Australia, and we obtained data points on where we know the insect is established. And we can actually use the climate data of where we know the insect occur to predict what are some of the new areas where this insect is likely to establish if it continues to spread. So the following is a map of South Africa with the black dots indicating the spaces or the areas where we know MFC is already established. The red indicates areas where it is likely to establish because it's a suitable climate. Now you can then overlay these red areas with macadamia growing regions. So it's highly likely that if we continue to move plant material, that MFC will also establish in other growing regions. So for this, our quarantine measures that is currently in place remains crucially important. Then we also looked at Africa in general. And again, I think it's important that MFC, if it is moved via plant material, is likely to establish in other macadamia growing countries. So having quarantine permits in place and restricting the movement of plant material to keep the insect out is very important. But more on the control side, we've started monitoring in macadamia orchards to see if there are any natural predators or parasitoids that can potentially be used to control MFC. I'm not sharing any data today because it's ongoing, but look out for more information potentially in the SAMAC journal later this year. However, how we have thus far used the data is to look at some of the species that we found, and we then went to see, are there any commercial products available to growers already, and do we see that they will have any effect? So what I'm quickly trying to show here is that we used two ladybug species, and for the second one we used both your adults as well as larvae, and we looked at how many MFC crawlers they can consume. I would like you to look at the last one, which is lacewings, and out of them our ladybugs compared to the lacewings, our lacewings work the best, and we do see that they have the potential to help control this problem. However, what we really do believe will help control is the Metaphicus macadamia. It is a specific MSC parasitoid. Of course, it was originally sampled in Australia, and then the Hawaiians have done quite a lot of research on it. We are lucky to say that thus far we've imported the parasitoid into the country. This is a project that is run by Anthony King at the ARC, but the picture that I'm showing you here is that we also now have a colony in our quarantine facility that we are using as a backup. We are just here to support Anthony, but Anthony has currently started with host specificity testing, and our long-term aim is we really would like to release this parasitoid to really help control MEC in South Africa. So this is my last slide to try and wrap up integrated pest management. Yes, the pictures is on stink bugs because it's our most economically important insect. But this is about us understanding the value of pest biology, continued monitoring, implementing cultural practices. We are focusing quite a lot is adding more into this whole biological control spectrum with the aim of reducing chemical control. Then, of course, I would like to just acknowledge SAMAC who supported all the research funding, and all the students at the University of Pretoria and FABI, specifically the Institute. And I hope that I'm available online to answer some questions. Thank you.